Hello all, my name is Sandy Wingert and I'm an environmental scientist at the Division of Water Quality. I have been with Division of Water Quality since 2007. I have my Master's of Science in Public Health degree from the University of South Carolina. I work in the Watershed Protection section and am in charge with restoring and protecting water quality in both the Jordan River and the Provo River systems. I have developed several TMDLs and watershed plans for this state, which not only outline the source and the magnitude of contamination, but also identify BMPs or best management practices to implement with the overall goal to restore impaired beneficial uses of surface waters. Today I've been asked to provide an update to you all on the E. coli TMDLs I am currently working on for the Jordan River watershed. So when I say Jordan River watershed, what I mean is the outlet of Utah Lake to Farmington Bay at the Great Salt Lake and all of the west side and east side tributaries. Originally, when I first started with this huge endeavor, there were 13 impaired assessment units or sub-watersheds within the basin. Each assessment unit would have their unique water quality or total maximum daily load analysis and implementation strategy specific to that area. Throughout this entire process, stakeholders have been involved and will continue to be involved. They are key players in developing these water quality studies. There are various sources of E. coli pollution within the watershed, and then at the end of this presentation, I will give you an example of what I'm currently working on for lower Big Cottonwood Creek. So what do? What we do, and why do we do it? Everything stems from federal regulation. So federal regulation regarding the quality of our nation's water began well over 70 years ago. The overarching goal is to restore and maintain the biological, chemical, and physical integrity of the nation's waters. They want all waters to be fishable and swimmable, in a nutshell. In 1972, the act was reconstructed to give authority over to the Environmental Protection Agency. Over the next 50 years, this regulation was amended to establish water quality standards, anti-degradation policies, to address long-term funding solutions for wastewater treatment facilities, and also to help beef up or establish programs such as the stormwater permitting, non-point source control issues, and pathogen monitoring for recreational waters. Note that Utah passed our own water quality regulations before the Clean Water Act was created. In 1986, Utah was delegated primacy over the Clean Water Act and associated programs. In 1991, the Utah Water Quality Act was passed, which created the Water Quality Board and gave that right to the director of the Division of Water Quality. So how does Utah implement the Clean Water Act? We do this through numerous programs within the Division of Water Quality. The first step that we do is that we define beneficial uses for our waters. One water body can have multiple beneficial uses, from drinking water sources to primary, secondary contact recreation, cold water, warm water, aquatic life, wildlife, and the class 4 is agricultural, and then class 5 is anything dealing with the Great Salt Lake. So for example, Big Cottonwood Creek here, we have it broken into two sub-watersheds or two assessment units. The lower one, so Big Cottonwood Creek 1, extends from the Jordan River up to the drinking water facility at the mouth of the canyon. Its beneficial uses are infrequent contact recreation, which is 2B, cold water aquatic life, and then agriculture. This was originally listed in 2014 for E. coli. Big Cottonwood Creek 2, so pretty much from the mouth of the canyon up to the headwaters then, has a different set of beneficial uses. So this one, we added in the domestic water 1C use to that as well. And because there's no agriculture up in the canyon, it's not protected for agricultural uses, so we don't have that category 4. This impaired use for E. coli was again due to the drinking water and the infrequent contact recreation use, and this one is listed on the draft 303D list for 2022, and I will talk more on that later on in this presentation. So the next step that we do then is we develop water quality standards and we monitor those. We develop water quality standards for all of our waters throughout the entire state. We establish criteria to protect those uses, those beneficial uses. It is also, these standards are reviewed and updated every 3 years through a triennial review process, which is also stakeholder driven. And then all changes are approved by the Water Quality Board. Note that the E. coli standards that we have for most waters in the Jordan River watershed, there's 2, 668 MPN, which stands for most probable number of colonies within 100 mils of water, and that is a not to exceed sample. The 206 MPN per 100 mils is representative of a geometric mean spaced over 30 days or throughout the entire recreational season. So not only do we establish the beneficial uses and establish water quality standards, the next step then is to monitor. We have been monitoring Utah's waters for decades, and everything that we base our programs on are based upon our monitoring data. So we can't do it all, and so we rely on cooperators like Salt Lake County to assist in data collection. All of our data is publicly available. So then after we monitor, then the next step then is that we assess. So when we do this, then what we refer to it as the integrated report, or the IR. This is a water quality assessment method that we use every 2 years. And it is like a conditional report card that we report back to Congress then, again, on the condition of our waters and any significant threats to our beneficial uses. That is compiled within a 305B report, and which refers back to the 305B section of the Clean Water Act. So not only do we do that, then we also look at the impaired waters that fail to meet the water quality standards set for those uses, and that is called the 303D list. I would be spending most of my conversation or most of my presentation talking about the 303D list and the ramifications of when a water body does get listed. So these are results from our latest approved list. We did a combined list for 2018 and 2020 integrated report, the IR. All of this can be found either in our report or on our website. So within this report, again, look at all surface waters, perennials, and canals across the state. And the assessment includes over 900 AUs were reported on. 80 of those were fully supported for their beneficial uses. 347 were considered impaired for one parameter or another. 32 AUs either have like a TMDL or a watershed plan in place. Over 80,000 miles were assessed. And for this current IR, approximately 2,000 monitoring locations were assessed, again, for rivers and canals. Okay, but what does that mean for the Jordan River watershed? So again, here you can see the boundaries, these AUs, these watershed, sub-watersheds that we talked about. And in 2006, then 28% of all the river miles that we assessed were considered impaired. The main pollutants of concern that we were looking at are temperature, salts, TDS, total dissolved solids, E. coli, pathogens, and dissolved oxygen. So in 2018 and 2020, 77% of all the river miles are now considered impaired. So this list has, expanded beyond just those field parameters, but now includes metals and more E. coli. I can see that my video is crossing out some of this, but so for E. coli impairments themselves, in 2006, we had 13% of the river miles were impaired for E. coli. In 2020, on this current IR, it has increased to 27%. And we also have the 2022 IR out for draft comment. And again, the findings on that actually have an increase in AUs within the watershed. So those impaired river miles actually increase about to 47%. But again, that's draft. So again, once it's impaired, if it's not impaired, we have an anti-degradation policy is set in place to make sure that those high category 1 waters, like the canyon waters that are protected, we implement measures to control that. We implement measures from the water quality studies that I'll talk about here next. And then we monitor, and then every 2 years we assess, and we have our marching orders as to move forward for those that are impaired. The one thing to note, though, that no matter if we are using various programmatic management options, I guess you can say, for these water quality studies, is that we take all types of sources of pollution into consideration, whether point sources, non-point sources, and natural background conditions. So when we talk about these impaired sites or these impaired water bodies and these water quality studies, then certain facilities, certain point sources will have permits. We can also look at the use. This is called a use attainability analysis. Then is that current use, is it appropriate? We can develop site-specific standards for water bodies where our default standards are not applicable. If the natural conditions don't need it, right? We can also go to direct implementation if there is a watershed plan in place that targets, or the implementation projects will be targeted specific to that parameter of concern. We don't even need to go to the TMDL route. But I will spend the rest of this conversation today on TMDLs. Okay, so when we refer to a TMDL, a division of water quality, it's two things. One, it is a calculation, but it also is the report itself. So when we talk about the calculation, a TMDL, total maximum daily load, is a calculation of the maximum amount of a pollutant that a water body can receive and still maintain its beneficial uses. It takes into consideration, load allocations from non-point sources, waste load allocations from permitted sources, and then again, that natural background condition. We also incorporate a margin of safety to address the uncertainty with our approaches as well. Okay. So this is an example of a very simple example. The existing load is what we find actually in the watershed, right? We're out there, we monitor, and when we say load, it is a multiplication about the concentration in the sample that we take times the flow of the sample that we take. So again, it's how much is coming in at one particular time. The red line is the water quality standard set at protecting at that use. And so what we need to do then is we need to decrease the existing load set at the water quality standard or the allocated load. Another way to look at this then, it's simply a pollution budget. It takes into consideration all various point sources, all various non-point sources, that MOS, that margin of safety we talked about. And then some TMDLs can also reserve an allocation for reserve capacity. And what that means is for any future sources that have not been earmarked during the time of the TMDL or future growth considerations. So again, it leaves a little bit of buffering capacity then within the allocation budget itself. So this is a typical TMDL process. Again, you'll notice that on the left, the stakeholder involvement process from start to finish. We start with the water quality problem. Why was it listed on the 303D list? The next step then is to come up with, source assessments. What is that? What is the TMDL target, which normally is set at the water quality standard? What is the linkage then between the impairments that we see in the watershed itself? What about the allocation? How are we going to break up that pie? Implementation and monitoring then, through permitting process, through reasonable assurance, monitoring plan and implementation plan. We submit the TMDL after it's been vetted and approved by stakeholders, by the Water Quality Board, and then finally to EPA for final approval. So again, it's a very lengthy process. So specific now to the Jordan River watershed. I talked about initially there was 13 impaired watersheds. We have the Jordan River AUs pretty much from the confluence of Little Cottonwood Creek downstream to Farmington Bay, so again, the lower Jordan River. Mill Creek lower up into about the Forest Service boundary. Little Cottonwood, Big Cottonwood Creek, again, through the valley up into the canyon mouth. Lower Immigration, Parley's Canyon, and then some west side tributaries from Rose, Butterfield, and Midas. Note that Immigration Canyon, it has its own TMDL for E. coli on that. It's the only one, well, I guess for the within the Jordan River watershed that has an E. coli TMDL present on that. However, this list was created part of our vision document back in 2014, 15, 16. And since then, as we continue to monitor, things are going to change. And so what that means then is that these impaired AUs have changed. The Jordan River 1, which we talked about, close to the Farmington Bay, is not, in itself, it's impaired. It's actually a tributary, North Canyon Creek, that's triggered the impairment. We created a new AU and a delineation, and this one will be addressed in a separate TMDL. In 2018 and 2020 IR, there was no new E. coli impairments for the Jordan River watershed. But however, on the draft 2022 IR, there was some new listings were added, and this includes Lower Red Butte, Upper Big Cottonwood Canyon, so pretty much the canyon to the headwaters, and Parley's Creek 2. So within this process, then we will determine then, do we incorporate these new listings into this into the TMDL report, right? This TMDL-wide, or would these be addressed in separate TMDLs, much like North Canyon Creek? So why do we care about E. coli? E. coli is one of several types of bacteria that normally inhabit the lower intestine of warm-blooded animals. Most E. coli strains are harmless. And again, we're talking about waterborne diseases, not foodborne illnesses or viruses, right? And so most are harmless, but some can result in gastrointestinal diseases. The reason why we sample for E. coli is, it's not feasible to sample for every single pathogen out there. And so we analyze E. coli as an indicator of recent fecal contamination. It's we get results back within 18 to 24 hours. They have a pretty good relationship built up between E. coli and fecal coliform, and it is a relatively inexpensive process to use. So moving into Lower Big Cottonwood Creek TMDL. So when I say Lower Big Cottonwood Creek, what I mean is the confluence of the Jordan River to the canyon mouth boundary. We have 4 monitoring locations within this AU. And again, most of this data that I am going to be talking on is being collected through a MOU agreement that we have with Salt Lake County. So you can see here that in this particular map, any of the red circles are impaired sites, and as you move upstream, they are no longer impaired, and I will dig into that a little bit further. So Big Cottonwood Creek at 500 West triggered the initial impairment. Again, when you look at this assessment, we only look at data collected during the recreational season, which is May through October. The first step then is to look at the not to exceed criterion. If you remember, it was that 668 number, right? So for 2B waters. And if we have more than a 10% exceedance of the 668, we say that that site or that AU is listed as impaired. So here you see Big Cottonwood Creek West, the original assessment, again, it was, we used data from 2009 to 2012 with about a 47% exceedance of that maximum criterion. Since then, though, we have moved the site upstream just a few hundred feet to the 300 West, and this is to correspond to Salt Lake County's flow gauge at that site. And here we can still look at recent data from 2017 to 2000. I think this goes through 2020 or 21. And we can see here that it still exceeds at a 33% exceedance rate. So again, we still have a problem. If we move up now to the middle of the AU, this is at Creekside Park. So this area is a stormwater detention basin from Salt Lake County. This is pretty close to the Holiday Lions area. In 2000, on the 2012 and 14 IR, this site was not impaired. However, again, we are always collecting data, and now on the current IR, this has a 26% exceedance rate. Okay, so now this is the highest site within the watershed, within this lower watershed. So this is Big Cottonwood Creek below the old mill. Again, this is a debris basin, detention basin for flood control. In the 2012, 2014 IR, there's no impairment. And then more recent data still shows that if you look on the left-hand side of the graph, it maxes out at 400 MPN. And again, there's no impairment here. So now we can kind of hone in then, instead of saying the entire AU is impaired, now we can start honing in and saying, okay, there's something going on between, that Creekside, between Old Mill and Creekside downstream then to the confluence of Big Cottonwood Creek. And here you can see that then. On the left-hand side is the Jordan River. On the right-hand side, hopefully they're not flip-flopped, is the canyon, goes downstream to upstream. So again, the Jordan River is on this side. Let's see if I can mark it. Over here. This site is the one that triggered the impairment, and then we've got the canyon mouth over here. And you can see that there is an increase in concentration as we move downstream. So one of the things that we look at then, okay, so then let's talk about sources then. What is specific with this AU? How is the land being used? How is the land covered? How does water flow in and out? These are looking at the watershed or AU characteristics will give us an idea then about sources, and not only the sources then, but how does it get from source in the loading pathway then, or the mechanism pathway, into the creek itself. So you can see here, and I apologize, this is pretty bad, but you can see here, this is land use and land cover. Anything red has to deal with developed. And we have here about 26% of high use developed. Pretty much the entire AU is covered with impervious surface, as we see here, and is fully developed. There's not agriculture. There's not a lot of agriculture here. There's not a lot of forest here. There's minimal amount of open space. So again, now it seems to be that when we look at this, we say, okay, in this watershed, we're not looking at agricultural practices or, wildlife that are triggering that, or at least, elk or deer. But we're looking at more of an urban source. And again, we're starting to hone in a little bit on the source assessment. So if we know that, then, but what about seasonality then? So this is concentration, when do we have problems with that, with E. coli? Again, at Big Cottonwood Creek at 300 West, this is the downstream location. You can see here that, that during the warmer months, then we have, right here, that's more of our problem, right? It exceeds our water quality standards, through late summer and early fall. And then at the Creekside Park, then, it is more numerous than, or it's more obvious than those exceedances happens in August. So then not only do we know spatially then something's going on between Old Mill and Creekside, not just, and then, but there's also now there's a timing, there's a temporal factor to this then. How was water being changed then between in the late summer periods? And this is where, knowing the watershed, talking with local stakeholders who have really a much more thorough understanding of the watershed is so important to help with this big picture of saying, okay, what is going on. So not only do we look at, the temporal issues, when do we have exceedances, we look spatially, we look at exactly when the watershed, but we also look at, how is water being moved in and out of these watersheds. And as you guys all know, that the Jordan River watershed is a highly managed system, with canals spreading all over the valley floor, taking Utah Lake water and bringing it into the farmers where, the drinking water facilities, then those providers then treat us with a higher quality of water and those exchange rates. And then not only that then, but you have, what's being captured then by the canyon itself, Big Cottonwood Creek Canyon, and those exchanges. And so again, water is moved in and out so much out of this valley. But it's still important to know that there is a seasonal aspect to all this. And then if you look on, but what about year-to-year variability then? In high runoff conditions like we had in 2011, we talk about loading flow times concentration, we're going to have a bigger pulse coming through during those types of conditions, especially if it's more non-point source in nature than we have in low flow conditions. But also too, you look at the month-to-month variation with these flows. Then, these mean monthly flows then, how does that play into these impairments and these exceedances of the water quality standard? So this is a table I took from EPA, looking at the relationship then between sources, potential sources and hydrologic flow regimes. And so what we use then is we look at a low duration curve, and I will get to that in the next slide. So here you can see, if we have exceedances during the high flow conditions, like the flooding events, the chances of that coming from a point source, so anything coming from a pipe, is very low. If we see it during those high flow conditions, those flood conditions, it's more like bank erosion, like overland flow, upland stormwater, unregulated stormwater issues. So in the inverse then, in those low conditions, those drought conditions, then it's not going to be so much precip driven like what we see with bank erosion or stormwater upland control. It's more going to be coming from a pipe because there's less dilution, right? And then between the moist to mid-range to dry conditions, you kind of have a plethora then of sources. And so again, we can use this then to looking at, that relationship between, when we have exceedances, under what flow regimes, where spatially do we have it, temporally, and now, looking at it hydrologically as well. So again, this is draft, but this is a low duration curve at Big Cottonwood Creek at 300 West. I need to update this with this more recent 2021 data, but you can see here that the red line is the TMDL line. So anything above that, we have problems. Anything below that, we're okay. And you can see here that under the high flow conditions, again, over here, we have a 17% exceedance. 17% of those samples exceed our TMDL. Under the mid-range conditions, it averages anywhere between 55 to 64%. Under the low flow conditions, we're at 40%. But if you remember, I'm going to go back one. I wonder if I can. Probably can. Okay. If you remember, then we've got reductions needed all throughout the flow regimes. Then, so that what that tells me then is that we've got several types of sources within this watershed. In the delivery mechanism, so point source in nature and non-point source in nature. So again, draft data, the same data that we looked at, but now we're just breaking it on a monthly basis then. So hydrologically, we need reductions in all conditions, all flow regimes, but what about on a monthly basis then? So the blue is observed loading. It's what we're seeing in the watershed. The TMDL, the orange, is where we want to be. That's based upon our numeric standards. So anytime you've got blue is greater than orange, we have a problem. And again, you can see that it's in those late summer months where we have less flow, maybe less dilution, or a change in the source of water, the delivery mechanism into Big Cottonwood Creek. So again, same data as I just showed, it's just in a tabular form now. Anything highlighted in orange is when we have our critical season, as you would say. And when you average that, then we're looking at about a 56% reduction in E. coli loading shared amongst these months. And again, this is draft. This is, we still need to update this data. So we go back to sources then, okay? So now we need a 56% reduction during those, June through October. The flow regimes tell us that we've got a plethora of sources, from non-point and point sources. And so this schematic was taken, I took from a TMDL from Wyoming. And I really like it because it gives a really good idea about what we're up against here. Is it direct deposition from wildlife or from avian, right? I mean, as the water moves down through the canyon, it slows as it reaches the valley floor. And, during Creekside Park that with the water slowing, then we have a more of abundance of avian and waterfowl. What about, what about failing septic tanks? Luckily, most of what's going on in here is not, most of it's on Seward. We do have some high up in the canyon that are still on septic systems, and that will be something that will be addressed when we start looking into the upper Big Cottonwood Canyon E. coli impairment. But again, surface waters, stormwater runoff control is another big one in this area, with how much impervious surface and how much urbanization this AU has. So again, when we look at this, then we look at all sources. So again, the regulated point source from treatment facilities to MS4s to on-site septic systems. Non-point sources, could be include agriculture, but at least within the Big Cottonwood Creek, that is minimal. Pets are another huge. Any open space or dog parks that are along the creek, that can be a potential source. We talked about wildlife, unregulated stormwater control, and what about people? Whether it's recreationalists or those who are unhoused. How does that fall into, or to possibly contribute then to these E. coli impairments? So implementation then. So now we've got the sources, potential sources, and again that will be teased out more through this TMDL process. Those are just examples. This is an example I took from the Immigration Creek TMDL implementation strategies. Again, the main goal is to reduce loading, E. coli loading from anthropogenic or controllable sources. We're not going to be able to control, where moose and elk, do their business. It's going to be, we need to control the sources that we can control. So some suggested BMPs or best management practices for Immigration Canyon, and again, these will be tailored for each specific AU. So these that are done or suggested on the east side tributaries might be a little bit different than on the west side tributaries because there's more of an agricultural influence up there. Or the canyons might be a little bit different than the urban, and because of the sources are changed, right? So again, any type of vegetation buffered, inventory and maintaining on-site septic systems, managing stormwater runoff, outreach and education, recreational management, sampling canals and ditches, and then also too with conducting DNA or MST, microbial source tracking testing. The Division of Water Quality issued a grant to Salt Lake County to conduct MST sampling, which they have done for the past 3 years all over the basin, all over the watershed. And we should be getting those final results then this winter. And how we use those then, it's something that the stakeholders need to discuss and saying, okay, then if we're not seeing, we've seen high E. coli levels in Big Cottonwood Creek, but we know that there's no dog presence up there in the canyon, well, then we can rule out and not spend our time looking into the canine aspect of it at all, but realizing, okay, then there's the likelihood of it being canine is less, but the likelihood of, people, if we're seeing high, human numbers, that's where we need to target. So again, it'll drive implementation, it will prioritize implementation. So I'm really excited to see those results. Okay, and like I mentioned too, that this is just, one example of several that we are currently working on. Again, a shout out to Salt Lake County for all of their monitoring efforts. We have over 50 monitoring sites well over the valley, main stem, Jordan River up through on both sides of the tributaries. So we will be spending the rest of the year, there we go, compiling this. And so the timeline then, in February of 2019, we had our first kickoff stakeholder meeting through the Jordan River Watershed Council. That kind of got set on the back burner as I was working on some TMDLs up in the Heber Valley. And then we started picking back up the pace again earlier this year, where we talked to the Stormwater Coalition. We introduced this very similar presentation to the Water Quality Board earlier this summer. And then this summer, then we finalized our technical approach. We're kind of going through the Big Cottonwood Creek example. This fall and winter, we'll spend writing the report and finishing up the data analysis. And we're hoping to have an internal draft done in the beginning part of 2022. And the ultimate goal then is to get this to the Water Quality Board then and off to EPA before October. So again, we've got several teammates working on this, Amy Dickey, Elise Hinman, Lucy Parm, myself, and Jody Gardberg, who's my manager. So again, thank you for everybody. And here is my contact information. Please feel free to reach out if you have any questions. I know this was kind of a lot, and yeah, so anyway, thank you so much for your time.